What's up, guys? Welcome back to Tidal Gardens. This is another episode we have of Reef Receipts here with Brandy, where we kind of cover some scientific journals and see how they apply or don't apply to the hobby. So, Brandy, thank you so much for coming back and joining us again.
Of course.
What are we talking about today?
So last time we were kind of talking about how corals interact with bacteria. So this time I want to talk about how bacteria interact with coral.
Okay.
So this specific paper is called Coral Mucus Rapidly Induces Chemokinesis and Genome-Wide Transcriptional Shifts Toward Early Pathogenesis and a Bacterial Coral Pathogen. Super long title, right?
Okay.
Doesn't really tell us much.
I'm sure it meant something to somebody.
So what this paper is looking at is Vibrio specifically. So when they say coral pathogen, they're looking only at Vibrio. They're also looking at how it behaves.
So Vibrio is a very common bacteria that's pretty much everywhere, but specifically it's kind of known, or certain types of Vibrio are known to be like really bad for corals.
Mm-hmm. So they basically say Vibrio is everywhere. And they were wanting to know how come sometimes it seems to be pathogenic and sometimes it's not. And it's just this new frontier of bacterial research that they're doing on corals. All this stuff is still really, really brand new.
Okay.
It's not something that there's a lot of research that exists on yet. There's a lot currently being done, but it hasn't quite hit journals yet. There's always a lag. I think this specific paper is really important, and it's a really good study because it's looking at the behavior of Vibrio, which is a very unique behavior, and I think could be important to us because if we know how it behaves, we might have some extra alternatives in dealing with it and maybe stopping it when it becomes a problem in our tank.
Okay. So before we get too much into it, they're looking at stuff like how pathogenic the bacteria is. So what is a description of like what does pathogenic even mean, really?
So is the bacteria going to kill you or not? Right.
Right.
So it's common. I think we've even talked about it in previous papers we've done where they found Vibrio, but it wasn't killing the coral.
Mm-hmm.
And so that was of a little bit of confusion to scientists because they think it should be killing the coral, but it's not killing the coral. So bacteria can have different levels, even within the same species, of being lethal or not.
Okay, so but how do you measure that other than just the stuff died?
The stuff died.
Okay. I thought we talked before about how like
with the UV.
Maybe it wasn't pathogenic then. It was like, like the about the growth rate and about how it overcomes defenses and stuff. Was that pathogenesis? Pathogenicness.
No, I think that was just what genes were on or not.
Oh, okay.
So
never mind then. Never mind.
Vibrio apparently is not always pathogenic. There are life stages of Vibrio where it's not going to hurt anything.
Mm-hmm.
It's just floating around, doing its thing, having a good day. And then there's going to be times that it's going to like just wipe out a coral overnight. And so that's the severity of the lethality. And so there are different genes that can be active or not active in Vibrio that is going to impact how quickly it kills that coral. It's still going to be really, really fast, but it's either going to be killing the coral or not.
Okay. Gotcha.
So one of the things that was really interesting is that Vibrio is kind of scary, actually. It's not interesting. Vibrio shows a different type of behavior than most bacterias. Vibrio does this thing called chemotaxis,
mm-hmm.
which some bacteria do this, where it can actually respond to its environment and move in the direction towards its environment. We don't typically think of bacteria moving intentionally to places. We think of it, it's on this table, and if I touch this table and put it in my mouth, I'm going to get sick.
Mm-hmm.
Not, it's on this table and it's going to move from this table to my mouth and get me sick.
Mm-hmm.
Vibrio does that.
Okay.
It also does this thing that's really, really scary, and not very many bacteria do it. E. coli actually does it, but it's a very, very rare phenomenon in bacteria called chemokinesis. So not only is it moving towards whatever it wants to be at,
Mm-hmm.
it speeds up. So
Okay.
it's like just lazing around, and then it starts like basically full on sprinting at you because it realizes that's what I want, which I think
Gotcha.
is really creepy. It's like zombies. Vibrio specifically likes coral mucus.
Okay.
So the thing that it's going to be running towards is coral mucus, and it's going to speed up as it sees that there's more coral mucus in the water. So this is actually really evolutionarily advantageous to the bacteria because it's not going to be coming into contact with coral very often. And if it's going to come into contact with coral, it wants to come into contact with coral that's already stressed out.
Mm-hmm.
And I think coral normally produces more mucus when it's stressed out. And we do know that coral do produce more bacteria or more mucus when it's already infected with bacteria. So it's like snotting, trying to get the bacteria out,
Mm-hmm.
or whatever the infection is out, just like our bodies do.
And so this is specifically about the amount of mucus, not necessarily the composition of it. It's just quantity of mucus.
Yeah. So it's reacting to concentration gradient. So the more concentrated the coral mucus is, the faster the bacteria is moving towards it.
Okay. So that kind of just made me think of like, there was, I don't know if it's very popular now, but for a while, people were especially in SPS aquariums, in Acropora specifically, they would intentionally like microbubble their tank, like aggressively. And when you do that, it makes the corals like slough off like a lot of mucus, and that goes to the surface,
Mm-hmm.
and it just gets like surface skimmed after that. So I was wondering if that was a bad thing because you're increasing the amount of mucus production on the corals. But anecdotally, people were saying that it works really, really, really nicely. So there could be like a lot of things at play, but that was something that popped into my head.
Yeah. I think I definitely have some thoughts on that, especially when you start looking at the time scale of how the changes in Vibrio starts to happen.
Hmm.
Like, I'm curious how long someone would be microbubbling
Yeah, like
and
hours.
hours.
Yeah.
It's
Yeah.
interesting. And they look better afterwards.
Anecdotally, see, this is the thing because I've never really tried it. Like this isn't my jam particularly.
Do they not do it very often anymore because only some people had?
I don't know. That's a good question. It's just not on trend. I think people have just lost interest and have moved on to the next fad or whatever.
Hmm.
But that was a thing that was talked about for quite a little bit as a technique in SPS aquariums to I guess like it's almost like a cleansing. You
Mm-hmm.
go to like a sauna or something like that and sweat it all out.
Empty
Uh,
the pores.
yeah. But now that I'm thinking, like, well, okay, so you've concentrated the amount of mucus production and everything like that. And theoretically, that would also then increase how the bacteria would then target that coral.
Coral, well, they're targeting the mucus.
Mm-hmm.
So if you're getting rid of the mucus, the bacteria might be following the mucus out through the skimmer, potentially.
Mm.
But if it's going on for hours, then I would imagine that the bacteria would be targeting down on the coral.
Yeah, perhaps. But at the same time, I think that if the coral is like sloughing off lots and lots of mucus, that mucus is then away from the corals. So potentially still good.
Yeah, maybe it has to do also with how sick the coral is, whether or not the slime is sticking around on the coral versus getting,
Mm.
or maybe the microbubbles gets rid of it. So this is like an interesting thing too, because they actually studied quite a few things in this paper. So they were looking at the speed specifically of Vibrio in when it's exposed to coral mucus and at different concentrations. But they also just wanted to make sure, just to CYA, what other things might cause speed to increase. So they also looked at the nutrients in the water.
Mm-hmm.
They looked at the temperature of the water because we know that bacteria tends to thrive a little bit better at higher temperatures. And then they also looked at oxygenation. And all three of those things cause the Vibrio to speed up as well. So
Okay.
microbubbling, I'm assuming, is increasing oxygenation.
Could be, yeah. I can't imagine that it's less from doing that to your water.
Didn't Andrew Sandler have issues with that?
Yeah, so this isn't something that hobbyists tend to have to deal with. This is like more of like a public aquarium thing, but I guess like Andrew's tank is as close to public aquarium as it really gets on the hobbyist end.
Probably bigger even than some.
Some for sure, but so okay. Rewinding a bit, Andrew was having some trouble, and specifically with his fish,
Mm-hmm.
right? So they were getting like Popeye and stuff like
Yeah.
that, and at this point, like Raj is helping him do a lot of the system design. Like this is recent, within like the last couple months
Yeah, he
or
posted
so.
it recently. Yeah.
Yeah. So what was going on was that like the gas saturation level of the water was very high, because in his particular system, like his aquarium is on one floor, but the sump is like a couple of floors down, like probably
Yeah.
like 30 feet down or something like that. And when he's moving the kind of water flow from the sump to the main system and back, it's like this cascading waterfall effect. So the entire sump basically is like acting like a protein skimmer. And there's a lot of gas exchange, a lot of micro bubbles. And that was oversaturating the water. And so I guess like the concentration of oxygen and everything else, nitrogen, whatever, in the water was so super high, that it was causing all these like health issues in the fish. And it's kind of interesting because there's another system that I'm familiar with from one of our wholesalers that Raj built.
Mm-hmm.
And he
I've been
was,
to this one.
yes, you have. So I don't know the exact volume. Let's say it's 5,000 gallons, something along those lines. But even though it's a smaller total volume than what Andrew's 17,000 gallon display is, the sump on that thing is probably like double or triple the size of Andrew's sump.
Yeah,
And
I'm fairly certain I sat there staring at it for 10 minutes and someone asked what was wrong with me. I was like, it's just huge.
yeah, it's this gigantic, gigantic sump. And I think that the skimmer sizes are roughly the same. But the reason why that sump is so big is that the the water needs to be slowed down so that there can be gas exchange, so you don't oversaturate the water. And I think that that's just one of those little aspects that are more well understood in system design at the public aquarium sized thing, something that Raj would normally have to deal with, versus like, oh, I'm setting up a 300-gallon tank with a 100-gallon sump. I have no idea what the gas saturation level of my water is. So yeah, that might be something that people aren't necessarily paying attention to for their home aquariums with
Yeah.
bacteria involved.
Yeah, because I think it's just the brain, how the brain works too, right? Like you see a pattern happening and you're going to look at the most easily seen things. Like I added this food or I did this. And that might not always be the case. It might have something to do with people who are having issues with STN or RTN. Maybe it has to do with sump size because the water's too oxygenated.
Mm-hmm.
Not
Yeah.
saying it is, but it's a factor in bacteria being pathogenic.
Yeah, and I think that there are like techniques that can be done to slow the water down, but this all has to be kind of like done intentionally. Like it's
Mm-hmm.
not just like you might luck into it, but a lot of times it has to be a little bit more intentional.
Mm-hmm.
So what are the other two factors?
Temperature.
Okay.
So bacteria becomes more pathogenic at 81 degrees Fahrenheit.
Okay.
And as in it's not pathogenic and now it is pathogenic.
Right.
So it increases its speed. It's looking for things. And then the other one was nutrient levels. So as it gets access to nutrients, it's going to naturally increase its speed.
Mm. That's So we here have gone to like keeping lower temperatures in general, like
Mm-hmm.
right around 76, 77. In the summertime, there's only so much that we can do. It climbs to like into the 80s, and I think that perhaps that could be the cause of like some problems. But for the longest time, I think that a lot of people were trying to keep their tanks between 78 to 81,
Mm-hmm.
and I certainly have found a lot of benefits to keeping my water temperature a little bit cooler.
Yeah. I mean, that's the sweet spot for where this Vibrio likes to start to switch on all of its pathogenic genes. So,
It's right around 81.
yeah.
Good to know.
Yeah.
Because I guess that's also part of like why we wanted to do this series, because it's like it's great to like look at the journal literature and whatnot, but sometimes it's like, so what do we do? What do we do about this? Like, do I just like run like a bigger UV and try to kill Vibrio, which is everywhere,
Yeah.
or are there like other like smaller steps
Controls.
you can take?
Yeah.
Yeah,
If
where
we
it's like,
understand its behavior and how it's reacting to different
yeah,
things,
it's
then
like you
we
don't
can,
necessarily need to like sterilize your water. You can just not have it be too hot, and then you're good.
yeah, like reduce the temperature a little bit. Yeah. So they also looked really heavily at genetics. So this is a really long paper, like 50 pages, which is very uncommon in a journal article to be so long because there's only a certain amount of pages they're going to typically give you to put the information that you want out there. Um, and they did a ton of studies and a lot of experiments were going on. So they also did a lot of genetic analysis. They were making sure constantly that they were actually looking at Vibrio because they're actually taking a camera and taking a video of the bacteria moving. And
Mm-hmm.
they wanted to make sure that they're actually looking at Vibrio bacteria, that it didn't get contaminated with E. coli or something else that we have around. And so they're constantly testing it. I think the lowest percentage they ever had of Vibrio was 99.4%, which means it essentially was all Vibrio, right? So they're actually looking at these bacteria and then they're doing the genetic analysis to say, all right, we're seeing actual physical differences in how they're moving based on these different things. What's going on genetically? What genes are turning on? Which genes are being turned off? So that was a really important step that they did. So what they actually went and did is in 2015, they went and collected 5 colonies of Passalopora.
So
And
this is science land. So this is Passalopora.
You're saying it wrong. It's Leviosa, not Leviosa.
Passalopora. Yep. And then they let it hang out in the air for 3 minutes because coral starts producing a lot of mucus because you're stressing it out when it's in the air. And they collected a ton of mucus from the coral. And then they cryogenically stored it, like doing all the science stuff, right, to make sure that nothing happened to it. And then they brought it back and started doing all these tests on it.
Mm-hmm.
So the graph, I'm assuming Becca can put it in somewhere. The graph shows what was going on with the bacteria when there was no coral mucus and then after they put coral mucus in with the bacteria. So the blue means that they're moving really, really slowly.
Mm-hmm.
The red and oranges means they're just zooming around. They got the zoomies. I'm probably moving too much. I've got the zoomies.
You're probably okay.
So they're moving around like crazy. And there's actually a video of this in the supplementary materials. I couldn't get it to open for me. I don't know if it's because I have a Mac or what, but I think it'd be really cool if I could get it to open.
It'd be hilarious that if we did find that footage and it's like, man, none of this is moving.
Wouldn't that be bad? Maybe that's why it's not able to open.
Yeah, just trust me, bro. They were moving.
I can't help it that the video's not opening for you. It opened for me and it was moving. Yeah, so the big thing though was like the gene expression. So they're moving fast. Okay, they got the zoomies. All of our animals get zoomies sometimes. But the gene expression, so what genes are actually active versus not active, changes 50%, which is basic, like over 50% actually, which is most of its genome. So it basically turns into a different thing altogether once it comes into contact with coral mucus. It's basically getting ready to infect a coral.
Mm-hmm.
So the ocean's really big. There's a lot of bacteria in the ocean. And if a bacteria cell happens to come across the coral, it's got to act really fast to get to that coral and actually be on that coral and then start replicating. It drops its flagella pretty quickly, all these kind of sciencey things that I don't know that people care about. But basically the time frame is within seconds it speeds up towards the coral. Within 10 minutes it has upregulated its quorum sensing genes, which means it's going to start becoming a sessile coral or a sessile bacteria. It's upregulated its infectious diseases. So its ability to break down cell walls and stuff has increased. It's increased its ability to metabolize. So it's not really using a whole lot of energy when it's out free floating in the ocean because it doesn't need to. But as it's getting ready to attack a coral, it's probably about to get a whole lot of nutrition and wants to start replicating like crazy, have lots of babies. So it starts actually using the energy to do these things. And then within the 10-minute to hour-long range of it being accessed or coming close to coral mucus, it starts to actually become resistant to different factors about the coral's immune system. They're called things like hemolysin, metal protease genes, things like that. So they're these really fancy words. But basically, they start to become resistant to the bacteria or the coral's immune system. So somewhere within that, the first few seconds of sensing coral mucus and the 10-minute range, that bacteria is in a state where it's probably able to be attacked in some kind of way on our end.
Really? In what way? That sounds like a very, very, very small window of opportunity to do anything.
It is. It's a very small window. But so it hasn't become immune to the coral's immune system within that 10-minute time range. But it's also increased its metabolism. So if you can somehow get it away from the coral so it doesn't get that nutrition input, it's probably just going to drain right through its health. And any stores it has is just going to be gone.
It kind of goes back to what we were talking about with the aggressive bubbling technique for those SPS. It's like, okay, so you've successfully increased all of this mucus production and everything, but the mucus is not sticking on the coral. It's actively getting sloughed off. So as the mucus is like sloughed off and it goes to the surface, all the I guess pathogenic bacteria is shooting up towards the surface away from coral, and then it's going to get dealt by like surface skimmer and your filtration and whatnot. So that could be just like an effective tool.
Yeah, because I mean, what's happening is the bacteria is using the coral mucus as a homing device to get to the coral. It can't tell where the coral is. It can just tell where the mucus is. And you would think that the higher concentrations of coral mucus would be coming closest to the coral. But if it's getting kicked out of the system, then you're breaking up that part of the behavior. It's not actually running towards the coral. It's running towards your skimmer.
And the nice thing is that it is it's a fast thing. Like it's not a 24-hour thing.
10 minutes.
10 minutes.
Because within 10 minutes, it's changed into a free-floating bacteria it's now sessile. It's like living on your coral as opposed to in your water. So it's looks like a completely different organism.
Right, because it's dropped off all of its ability to swim, and now it's really kind of like setting up shop to like replicate on a surface.
So it's lost its flippers. Some of the weaknesses in the paper. So there are some weaknesses in this paper, but the weaknesses are, I think, necessary. I think that they decided to do this intentionally because this paper needed to be published. This was something that we were talking about. I don't think people always realize that there's some tricks that have to be done sometimes to publish a journal. And I've had to do this before, right? So you have data that needs to be out there, but if you don't hit certain numbers for significance or whatnot, they're just going to immediately reject you. So there's going to be no publication. And it's not that, I know a lot of people think that some of this stuff is like, oh, the people who know each other, the powers that be. But all of this is blind reviewed. You submit a paper, the the review board has no clue who you are. There's no identifying information. And if there is...
They don't know your institution. So it's like, oh, this came from Stanford. It must be good or whatever.
Right.
There's none of that.
None of that. And if you happen to be friends with someone and you're like, oh, my friend is doing this study, you should go and say, hey, someone else needs to review this.
It's like a professional ethics thing. It's like, I'm going to go ahead and recuse myself from this. I know Jamie Craig's sort of thing.
Of course, there's so many people doing this and they're getting so many papers. The chances of that happening is very slim.
Sure.
But I have had a paper come to me once that I was reviewing, and I was like, I'm fairly certain I know who this is. And I shot the editor a email, and they're like, yep, we're going to give you another paper. Like, so it does happen occasionally, but I've only ever happened to me once. But I do think that they did some things that their data isn't saying what they are telling us it says for some of these things. So their supplementary materials are probably 3 or 4 times longer than the actual article. And a lot of their supplementary materials, they walk back what they say. So in the article, they're very confident. This X means Y. And in the paper, they're like, we think X means Y. Or in the supplementary materials, they say, we think X means Y. So they walk it back a little bit. And some of their data, just what they say it says, I'm looking at the data, the graph, and I'm like, that doesn't say that. So
That reminds me of like football, because we talk about football just a little bit.
yeah,
And in celebration of the University of Michigan Wolverines winning the national championship,
congratulations.
let's talk about CTE. So apparently, like the CTE is rampant if you play football. Like basically, if you play at
That's what the media tells us.
I think CTE rates are crazy high. Like if you
Oh,
play
that's true. Yeah.
high school or college, it's like 90-something% you're gonna get CTE.
Mm-hmm.
But it's a lot less clear how CTE negatively affects people. It's probably not great that you have brain damage, but there was a research study by this Dr. Benjamin Omalu, I believe.
I never saw this movie. So.
Well, and he was the one who like coined the phrase, something encephalopathy. It's CTE, right? Chronic.
I don't know what it stands for. Encephalitis is
Yeah.
I can't even pronounce it. I would need the words in front of me.
The brain damages.
Yes.
And in the movie would make it sound like, okay, if you get CTE, it's like a death sentence. But
And not just a death sentence, like you're going to kill yourself.
yeah, it's gonna be bad.
And we were talking about this because I was like, you would think that there's a lot of suicides with football players then. And I just went and looked at the data.
And there's not.
Very few suicides.
And if there are suicides, it's not necessarily has anything to do with the CTE part of it.
Yeah.
But you have this movie that's out there, and that's Will Smith, taking on the NFL, like uncovering all these truths, and going back to like the supplementary materials thing. It's like when you actually look at the original research by that doctor, it's heavily criticized. Heavily criticized. And there's this also happens in law a lot, where you might see like a very punchy headline about like some court case, but now I know, because having gone to law school, it's like I know if I see any journalistic thing talking about a case or the law, I can pretty much not pay any attention to it, because there's so much nuance to it. And there's a really good chance that whatever happened as involving that case, that court opinion literally might have every other line criticized by another court, to the point where that law is so weak and so bad that nobody would ever really follow it. But you have a journal article with Will Smith, and suddenly it has like it's designed to sell advertising.
It's almost like you have to go to law school to understand the law.
Yeah. and you know what? I kind of still don't.
Yeah, it's complicated.
Yeah. So it is kind of interesting where like even though there is a research study out there, there are some very valid criticisms of it just built into that.
Yeah, so like some of the data that I thought was problematic was they were talking about the speed of the corals. And when you look at the graph and you look at that, so this was one thing I was taught whenever I have very well trained in statistics. I like numbers a lot. The first thing that you're taught to do is look at the data. You don't start doing ANOVAs or t-tests or anything. You look at the data to see what's going on, because there's certain assumptions that have to be made. Like, is it normal? Is it heteroscedasticity, which is like how evenly spread the data is. Like, there's certain things that numbers have to be somewhat similar in order to compare them in certain ways. And if they're not similar in those ways, then you need to use a different test. And a lot of their data is not similar in those kind of ways that is appropriate for the testing they used. So the testing that they use, I can look at the data and say, yeah, it's going to say that there is no difference between these two populations. But looking at the data, I know you need to use a different test because the data is, or the populations are different, because the heteroscedasticity is so different.
Right.
So,
I totally forgot about that. Like, going back to like in like college years, it's like certain statistical tests only work in certain situations, and if you do that test in not ideal population conditions, it basically doesn't work. Like the numbers and conclusions you get back are just not particularly strong. There's not a lot of validity to it.
Yeah. I mean, so heteroscedasticity has to do with how variable the data is, and how it changes. Like, is it getting more variable as it gets bigger or smaller as the time goes by, whatever the variables are. And that needs to stay the same. If that's changing throughout whatever your axes are, your analysis is just not, it's not telling you anything. It's like adding apples and oranges to figure out what the weather is going to be. It's just completely not valid. One thing does not mean the other. So a lot of their data wasn't the test I would have run. But I don't know that the paper would have been published if they had used the tests that were more appropriate. So I kind of think that the weaknesses may have actually been intentionally done so that it would get published. And I'm okay with that, because this paper, I think, has a lot of really valuable information, and I'm okay that they played the game to get this published.
Corruption. Overall, a good paper?
Very good paper. I think they did a lot of CYA stuff, right? It wasn't just, I'm going to go do this one experiment. They were constantly validating and double checking that what they thought they were doing, they were actually doing. And there's nothing wrong whatsoever with the methods, and anything that I thought, oh, it would have been nice if they had done this. They had already done it, plus some. Like, they did more than even I would have done if I was designing this experiment.
And so the criticisms are more on like the analysis tools at the
presentation of the data.
Yeah.
Yeah. Though I don't think it changes the outcome and like what the data means.
We'll have to see the video to see all these completely
see if it's actually moving.
Stagnant corals or stagnant bacteria.
Yeah. So that was one of the things, right? The bacteria, they determined if a bacteria was living or dead based on how much it was moving. And they were like, oh, there's no difference in these things. If you look at the data, there's difference. So I'm like, I don't know how much of the bacteria was actually dead or not, because their numbers don't seem to match what it looks like the data would say.
Okay, so breaking that down just a little bit further. So they're saying that dead bacteria isn't moving around.
Right.
So we can ignore it.
Yes.
But if there's live bacteria that's not really moving around, that should not have been kicked out. That is like counter to their thesis that the bacteria should be moving more aggressively. And because you have these bacteria that are not moving, it's like, oh, those are dead, guys.
Yeah.
Let's pay attention to these fast-moving ones only.
Yeah.
And therefore, we have like a very, very, very strong data set that says that you do this, the bacteria go crazy if you ignore the ones that aren't.
Yeah, and there's a huge dip, actually. When you first add the coral mucus, there's a huge dip in speed of all the bacteria. If you look at the supplementary materials, this isn't in the paper. And then it like gradually comes back up. But like you said, they're ignoring the ones that aren't moving, so it's not being included in what's going into the actual publication that the reviewers are reading.
Right.
And again, I don't know that a reviewer would have read if they had put the data that was in the supplementary materials, if they would have published it. I appreciate the heck out of them that they included the supplementary materials, though.
Nice.
They did not have to add all this stuff that was like, but go look at this data.
Nice.
Way to
Very
get
cool.
around it.
Yeah. So, guys, thanks for joining us for this latest episode of Reef Receipts. And that's why we check the receipts. We got to see what's there, right? So, yeah, guys, I appreciate the viewership. And thanks again, Brandy, for coming and hanging out with us.
Thanks for letting me do this. This is so
Yeah.
fun.
And until next time, happy reefing, y'all.
Bye.